Hello, everybody. It's a pleasure, it's an honor, it's a privilege to be here. Alison, thank you for the invitation. I am a clinician and a scientist like Jamie, and actually following Dr. Martin and Jamie, I may not have very much to say today, but I will walk through how we think about this idea of autism, what autism really is, and when it's helpful as an idea, and when it may not be as helpful as an idea when we think about how we are trying to impact the lives of kids and youth and adults who identify with this particular label. So this is me. I'm a clinician scientist. I have a couple of disclosures for you there. I do receive funding from both federal provincial sources in Canada, but also some industry, and I have a couple of patents. They don't impact this talk, but if you have questions about them, I'm very happy to answer. So again, a lot of this has been discussed, but if you allow me to kind of follow my train of thought through to make the case. So as a clinician, we see kids who identify as autistic or having autism in clinic all the time, kids and youth and young adults in my case. But they almost never, unless it's at first entry point where they're looking for a diagnosis, they almost never come to clinic because they have differences in social communication abilities and repetitive behaviors, which is how we define autism, right? They come in because they have difficulties with what we consider usually mental health domains, so anxiety and ADHD and obsessive compulsive disorder and mood disturbances and irritability and aggression and self-injury, or they come with complaints that usually go to the child neurologist like me, which is sleep and epilepsy and sensory motor dysfunction, or they will come in with other common pediatric problems that are persistent, like gastrointestinal distress or immune differences. We also note that the kids, as they are growing up and they are accessing the healthcare system, they have a variety of abilities, right? So we have people who have thinking skills and learning abilities and communication abilities that exceed that of an average person. There are colleagues, our doctors, our lawyers, but we also have people who have a lot of difficulty learning new things. We have people who find it very difficult to use verbal language to communicate. So that's so, and thank you for the introduction. I guess I've said this too many times, but it's possible to find a kid with autism that's more similar to another kid who carries a different label than two kids with autism. So if you have a kid with an autistic child who does not have intellectual or thinking challenges or learning challenges, communicates with verbal language really well, but has severe ADHD and anxiety symptoms, you can imagine how clinically that looks more similar to a child that does not have autism, has ADHD and anxiety and maybe a little bit of social deficit, than an autistic child who has severe intellectual disability, irritability, aggression, and they self-injure, right? So clinically, the label is not helping us think about these kids. The last two kids are more similar. Clinically, we perceive the first two kids are more similar. The other thing to think about is that kids come in with a variety of associations, if you like. So some of them, and I say kids, but think about kids, youth, and adults, many of them will have a genetic syndrome or a genetic variant that we think is highly associated with the pattern of strengths and challenges they have. Some of them will have a pre-existing medical condition that highly increase the likelihood of them having a pattern that is what we call autism. Things like congenital heart disease or severe prematurity. We know those kids are much more likely to present with the autistic label than kids who don't have these conditions. And a whole lot of other kids and youth and adults have no obvious association that we can think of for very similar types of presentations. So as a clinician, when I put my clinician hat on like Jamie and I go to clinic, I see many, many, many very distinct conditions clinically that we all label under autism. And I see many kids who carry different labels that look quite similar clinically in terms of their profile, of strengths and weaknesses, but also their needs. So then the next question is, does biology help us clarify any of that? Can we use biology to understand all these different autisms? And can we use biology to understand how these different autisms overlap with all these other neurodevelopmental, mental health, and physical health conditions that the kids present with? So again, following Dr. Martin, this is not going to add much, but tons of investment in genomics over the years, all over the world, by many agencies, many groups, enormous amount of commitment and investment in this field. I'm just going to highlight a very recent paper where Dr. Steve Scherer's group from Toronto put together all the genomic variation that has been reported around the world by multiple groups to give us a sense of what we know about the genomic architecture, if you like. How the different genes, the types of different variations in the different genes may be contributing to what we are calling autism. And this has a replication cohort, so we use Missing, which is a large repository of Canadian, American, and international data, and then replicated to the Simons cohort that you just heard before, for a total of more than 11,000 participants in the study. And to say that when people focused on these very high confidence genes or variants, the things that we return as a clinical finding to the family, not just the things we're interested in, there were 134 genes this time around that met that criterion. So if you think about my question about how many autisms we have, that would say you have 134 genetic, highly confident genetic versions of autism, right? A little bit daunting, obviously, if you think about what a clinician would need to do with that number of conditions and how we think about treatment or intervention. But also, if you look on what I think would be your right side of the screen, those did group, as they have in the past, in certain groupings that reflect common mechanisms, so that hopefully we're not talking about 130 or 140, but we're talking about 5, 8, 10, 15 biological versions based on mechanism of autism. However, no genomic variant in this whole collection is specific to autism, right? So these genomic variants increase the likelihood of developing autism, but they also increase the likelihood of developing a whole bunch of other things. So this is just an example of from the Baby SIVS Consortium, from the Canadian part of the Baby SIVS Consortium, where we looked at if a family has a genetic variant, and we know what it is, right? And we looked at the chance of the second child having autism and having the same variant, only less than half the time we could predict the diagnosis of autism in the second child. However, if we were interested in all those symptoms and signs on my first slide, any neurodevelopmental difference, then our ability to predict that the child will have some neurodevelopmental challenges jumped from 46% to 85%. Again, telling us that this biology, these genetic variants, are predicting a group of neurodevelopmental differences, but this, I think, that we call autism, genetics has a problem predicting. So the question then remains, for all the investment we've made into genetics, are genomics adequate for us to understand all these different autisms, all these different biological categories, so we can offer kids personalized treatments? And I think based on what I told you, I hope you agree that, the other thing I did not mention is that the percent of the group, and that was a question before, that had a clinical variant, a genetic variant that you could return as a clinical finding, was only 14%. I mean, 14% is not a small number for those of us who kind of play with genomics, but it is a small number. So if we think that only 14% at this time, we can give a particular information about their genetic variant, and that genetic variant may not predict autism, but any neurodevelopmental difference, maybe, although genetics is critical, it's the blueprint, it constrains the possibilities of outcomes, it's just not adequate. So we need to also understand our brains and our bodies. And so the example I'm going to show you in my last 2 minutes is then how we moved from thinking just about genetics to how we link from genetics to brain and body differences to try to understand different groups of autism and how these different groups of autisms overlap with other neurodevelopmental conditions. So Jamie talked to you about the ABCCT, which is a large American biomarker consortium, and I'm going to show you data from the PONT network, which is a Canadian consortium that takes the approach that all of these neurodevelopmental conditions have a structure to them. They're overlapping. How about if we put all the kids together and start understanding how genes affect brains, how brains affect behavior and thinking skills, and how we can use that information to get kids that are more alike in their biology together rather than what's predicted by the diagnostic label that we gave them, right? And it may be that the diagnostic labels will work very well, and in fact, when we ask the brain to tell us which kids are similar, we're going to get an autism group and an ADHD group and an intellectual disability group, or it may be that, as our clinical experience would suggest, this would get messier than that, right? So there's 4,000 kids right now in the consortium in terms of the numbers I'm going to show you. It's a largely co-designed consortium with families and youth and adults of a variety of neurodevelopmental differences. Okay, so the first thing we did is we assumed that our labels would work. So we looked to see whether kids who have different neurodevelopmental diagnostic labels actually have different brains. So we compare kids with autism, autistic kids, to kids who have ADHD and kids who have intellectual disability and kids who have obsessive compulsive disorder and a variety of other conditions. And the take-home point for this one, I think from your left to right, you're looking at how the brain is connected structurally, how the brain is constructed in terms of its thickness and its volume, and how the brain is functionally connected, so how the different parts of the brain talk to each other. We found that on average is a critical word here, the groups looked about the same. There were some small differences here and there, but on average, the groups looked about the same. We could not distinguish on average with large enough samples. However, they didn't all have the same brain, and that's the critical piece. There were many different brains here that we were averaging in each diagnostic label, but the diagnostic label did not help us figure out which kind of brain you had. So then to solve this problem, this is how we use mouse models. We were to our colleagues, Dr. Jason Lerch and Jacob Elligood, who scan mice. And so we had mice made with a variety of those autism-specific and some not so specific neurodevelopmental variants. And we asked them to scan them. And then the idea was to try to learn from the effect of those autism genes on brain structure and function in the mouse and take it back to human. And apparently I'm already over time, so you're going to give me another minute and a half. All I want you to do is just focus in the middle blue chart, where on the x-axis, so on the bottom, you see a variety of models, genetic and otherwise, of autism. And every column, every blue column is one of those genetic or otherwise variants. And then the difference from the zero line is how different you are from a typically developing mouse, if you like, right? And so you'll see that some mice have really, really big brains on the right side. Some mice have really, really small brains on the left and everything in between. And so if I had taken an average again, I would have told you the group is a little bit different from typically developing mice, but there is a lot of variability, which is what I told you about the human condition, right? But it's not noise. It's not some random, some people have big brains, some people have small brains. It's that the genetics, the biology of that particular mouse predicted where they're going to end up on that curve, right? So we are learning that the variability has biology to explain it. And because of that, we went back to our human data and started clustering using artificial intelligence methods, started clustering the brains across these neurodevelopmental conditions to understand how many different types of brains we have, assuming now that they have biology that predicts where they end up on these clusters. The one thing I want you to notice from this slide is that, let's say on the top graph, so you'll see 10 clusters. It doesn't really matter what this is. The point is there are many clusters, almost none of them are specific to autism. So different kinds of different autistic kids group with other conditions. And if you look at the left where the yellow bars are, a bunch of autistic kids actually look pretty typical in terms of their brain development. They group with a typically developing kid. So we have autistic kids who seem to have neurodevelopmental brains, and we have autistic kids who seem to have almost typically developing brains, right? So then the next question from that is, what predicts whether you're going to get a neurodevelopmental brain, and what predicts whether you're going to get a more typical brain and still have the label, right? And so in this case, we are replicating now on a independent cohort, which is HBN, which is a New York cohort out of ChildMind, so it's your population. We are asking machine learning methods to start dividing functional connectivity, so how the brain connects, to different groupings of kids that have similar brains. Again, I can answer questions about this, but the lessons learned are the most impactful thing on the type of brain you have is your cognition, so your thinking skills. That's the one that has the largest effect. After that, for us, was ADHD symptoms. And we didn't get to autism symptoms much later in the details on top of everything, right? In fact, socioeconomic status had a bigger signature on what group you're going to end up than whether you carried an autism, whether you had autistic symptoms. So how are we to interpret this? We're to interpret this that the way the brain understands categories is not the way the DSM understands categories. And to the task I was given by Alison, how we think about autism may need to be rethought. And consistently with what has been said before me, the many different brains and the many different biologies and bodies of autism may need to be rethought. Why? Because I put this from the Lancet Commission. Even in the Lancet Commission, where we didn't deal with biology at all, we talked about personalized health treatments. Well, how do you personalize a treatment if you do not know the condition the person has, right? So understanding these clusters, their biology, and how they contribute to the different brains and bodies is critical to personalized treatments. The last comment I'm going to make, because I got to stop, is the experiences of people who identify as autistic are also quite varied, and they're not stable. So this is an example where in Canada we did a very large prioritization exercise with our stakeholders about what they would like to prioritize as a target for research in treatment and intervention. I can go back for the specifics if you have questions, but the point is, with the exact same methodology and the same moderator, the UK came up with slightly overlapping, but almost completely different list. So it's important to know that I'm not taking this to say that they're both very valid. Different people have different versions of autism biologically, different ideas about what the autism means to them, and different areas where they want intervention. And understanding that our job is to meet the needs and the wants and the preferences of the autistic people wherever they are on these spectrums, whether we're talking biology, needs, or preferences, is a critical point in the debate about where intervention research goes, but also critical on how we think about autism as a construct. So, summary, many different autisms. We know that from clinical experience. We know that from biology, whether you take genes or brains, all the other body systems I didn't talk about, many different experiences and needs and preferences about what should be treated. I think the challenge for us is to actually reconceptualize, and I would agree with Jamie on this one, of how we think about what needs to be done in autism research, with the ultimate goal not to move some autism idea or some autism construct, but meet the needs of people who are actually asking for intervention work. Part of it is a large computational exercise. I hope I can convince you of that. So sharing data, collaborating around the world would be critical, not just in genomics, where there's more experience of that, but across all the other things that we do. And then I'm just going to say thank you to the many, many, many colleagues and families and the youth and the families that co-design and the funding agencies that contribute. And I'm going to stop at that.
Do we want to start on this one? Okay. I'm over to your left. Hi. So I had a question. How do you think our understanding of the heterogeneity that you were talking about in brain biology will impact diagnostic criteria moving forward?
So I can say something for the short term and something for the long term. So for the short term, actually, I'm going to go the other way around. The long term, this is like the ideal state. Ideal state, we have some clarity about the categories that predict the kinds of needs that the kids will have as they grow up, not just their presentation right now, but what trajectory they're on, right? So we can plan to provide the skills, the medications, wherever you're comfortable. I'm comfortable in both spaces that are required to optimize the kind of version of good life that they want. We're all working for our version of good life. Our version of good life is a personalized decision. If we have an idea of what range a child is on based on their biology, I'm hoping that ultimately our criteria will map into that. There may be behavioral, there may be biological. I'm a neurologist, I'm not a big behavioral criteria person, but I do defer to the experts in the room around that. But the idea is that what we have now does not predict outcome, right? Does not predict trajectory. So I'm hoping that what we're going to get to is a system that allows us to predict trajectory. In the short term, I think there are a few problems to think about, and we need to be honest about that. It's not trivial. So first of all, we classify for all kinds of reasons. We don't just classify for figuring out interventions and trajectories, right? So people find community in these classifications, they find identity, there's all kinds of other complications that are important to take into consideration. Secondly, we have built our healthcare sectors around it, and so although I do believe all healthcare sectors are inequitable because they are built around these labels, I do think we cannot trivialize what it would take to revamp the system to actually be able to provide services based on profiles and needs rather than a diagnostic label like that. But I also think we need a bit of humility, right? So there's lots of things, like we know what we are starting to get comfortable with what we don't know. But it's not like I propose to you an alternative classification system, right? That we are far from saying this is not working and this is what should replace it. All we're saying right now is this is not working, right? So it's important to think about a little bit of humility as we are planning systems and sectors to allow for flexibility as we're going to learn more. I think biology will be important at classification. I do think some markers that tell you what predicts that have big effects on brain biology probably should be part of that discussion and classification, whether it's cognition or something else. But we still need a bit of humility. Like the worst thing we could do is replace a bad system with another bad system, right? That doesn't work.
Thank you. Your concepts are very interesting, but I couldn't read your slides, and I'd like you to answer, what are the top 3 priorities for the individuals in Canada and the top 3 priorities for the individuals, the group in London, and any comment on the differences?
Yes. So you will notice that the Canadians are a bit more biologically based in their priorities. So although they did include some family support or family training, in their top 3, they have 2 precision health priorities. So they want to know what, for whom, when, for how long, what's the dose. So they want precision solutions They don't get into specific symptoms until, I think, number 5, and then their priorities are anxiety, irritability, and emotion regulation, anxiety, irritability, and lastly, at number 10, the first time you see a core symptom domain, which is social skills. Well, for the UK people, and yes, those are actually published. You can actually find them on the website. You'll see that there is a lot of supports and a lot of mental health, which again, I'm not surprised, but precision health doesn't show up very much. Biological treatments don't show up very much. And core symptom domains do not show up at all. So they represent slightly different perspectives. Now, just to be clear, the top 10 came down from a top 1,000 something. Right? This is a standard process that happens over a year of systemic iterative communication back and forth to narrow the list. So these are not the only things that are on the list, but these were the top 10. Yeah.
Hi, first of all, I just wanted to say thank you so much for all the research that you have done for people with autism. My question relates to mortality and specifically premature mortality. The reason I ask this is my understanding is that people like my son who have autism and other disorders such as epilepsy and intellectual disability are the most likely to have the shortest lifespans. How does this factor into your research? Thank you.
Yeah, that's a really, really good question. And it gets to this idea about why you need to know about biology. So again, I'm not going to talk about my data because this cohort now has grown up to young adulthood, but we don't have yet middle adulthood and aging adults in the cohort. But my interpretation of the literature, as you said, is that unexpectedly, we have higher mortality in this population than we thought about. And if I think as a neuroscientist or as a neurologist rather than as a behavioral person, a lot of these genetic variants have multi-system involvement, first of all. Secondly, they predict other medical conditions like epilepsy, which is very prominent, that has its own mortality and morbidity profile. So understanding the biology of the child that's in front of me, rather than giving them the label of autism, allows for this thing that we call anticipatory guidance, thinking about what does this biology predict for the child, not for the label, but for the child. Like, lots of our genetic variants involve heart, involve kidneys. Lots of our genetic variants are associated with severe forms of epilepsy. Lots of our genetic variants predict regression in adulthood. Knowing these things will help us provide the health care, and I know it's not all about health, but I'm a physician, right? Will allow us to provide this personalized health care that will optimize the outcomes for the people that we serve the way they defined an optimal life, right? So that's why biology is important, because you are right. Like, I can think of several of variants that are associated now with earlier death than predicted, and knowing that would have been a useful information for the family, for the person, to plan, to think about interventions, to invest research, and so on. I'm good? Perfect. Thank you.